وریا is a young faculty member of Niels Bohr Institute, University of Copenhagen. His research field concerns the application and development of computational microscopes. Very recently, he has developed a method for simulation of realistic size cellular organelles. Today, he will talk on the spatial organization of biomembranes. Uriya, thank you very much for accepting our invitation. Please start your talk. You have 45 minutes.
Yeah. Yeah. Thank you very much, Dr. Najafi, for the introduction. My name is Uriya Pezeshkian, and I am going to talk about part of my research interest, which is computationally investigate biological membranes. I do often collaborate with experimentalists, but I don't do any experiments, and I do theory as well, but it's not my focus area. So this talk is organized in 3 parts. In the first part, I will give you an introduction about biomembranes and interesting research questions or research area that we can usually, as a physicist, enter. Then I will talk about the state of the art of computational methods, the methods that I am using, and the perspective for these methods, so what would happen in a few years, what we expect in 10 years, for example. And then here also in the second part, I will also talk about some of the cellular processes that I use this computational method to investigate. But I will only briefly mention them. But at the last part, I will more in detail, I will talk about one of the recent work that we have done in a collaboration with the experimental groups, which to creating synthetic membrane shaper for to create a division machinery for artificial cells. Okay, before going any further, I think the concept of hydrophobic effect should be clarified because they are everywhere in biological systems. So, in nutshell, you can say some molecules or a part of a molecule just hate water. This and then in opposite to this effect, so you have a hydrophilic effect, which eventually the one that like water. This effect has an anthropic nature and is often seen as perturbation in the hydrogen bind dynamic of the water molecules. So it's not the hydrogen bonding itself, but the dynamic of it is get perturbed by this molecule because they can't form hydrogen bonds, and therefore this molecule loses entropy. Water molecule loses entropy, and therefore pushes this molecule, all the hydrophobic molecules, or into a region, force them to self-assemble to minimize the contact with these molecules. So a lipid is a molecule that has a hydrophobic part and a hydrophilic part, which usually are referred to as amphiphilic molecules, but here my concern is lipids. And phospholipids, for example, are very common type of it, and here is a very good representation of, or maybe schematic view of a phospholipids, that you see the head group, which is hydrophilic, likes water, and a chain, which is hydrophobic, dislike water. And these chains are typically 2, but you have cholesterol, which is 1, or cardiolipin, which has 4 chains. And also this king here actually is very important. This represents that there is a double bond in this carbon chain, which is important. Otherwise, in a physiological temperature, these systems often exhibit gel phase instead of a liquid phase. And when you have these molecules inside water, they self-assemble. They form micelles in low concentration. Then they can form liposome, which if they grow more, it will look like a bilayer sheet. But eventually the edges never can be stabilized, so always the system closes up. There are many other structures that they can self-assemble too, but my concern here is only the bilayer sheet or liposome. But these pictures are very misleading actually, because membranes are not like that. They are flexible. They fluctuate a lot. For example, this is a fluctuation at the nanometer length scale, while this is a fluctuations at the micrometer length scale. And you can see that although the size can extend up to micrometer size or even more, hundreds of micrometer, while the thickness is still only 2 molecule thick. So make them very interesting. And they are fluid, so then they can adapt to different shape, or they are not a static structure. And the way you see them in experiment, usually this is from fluorescent electron microscopy, that you see such a line actually. So that is what you observe in experimental settings. And then this is a schematic view of all membrane in a eukaryotic cell. So you could see they exhibit a diverse range of shape, from almost planar plasma membrane to highly curved shape of smooth ER or Golgi operators or mitochondria. Not only the shape is complex, but also membranes are very complex in the composition. Here is a segment of a plasma membrane that is obtained from a computer simulation. And you can see that they are highly packed with membrane proteins. So although they are still a lipid bilayer, because the main components are lipids, but this protein really disturb this bilayer nature. They still share a lot of common feature, but they are not the same. And when I usually refer to biological membranes, I refer to something like this that has protein in it. While lipid bilayers are only made of lipids, but maybe multi-component systems. So why do we explore biological membrane and membrane shapes? Is that to understand cellular processes, many cellular processes require the formation in the membrane shape. Actually, membrane is an integral part of these processes. So to understand them, we have to have a mechanistic understanding of biological membranes. Also, nowadays, we are trying, as a scientific community, we are trying to engineer a cell from bottom-up approach. So one of the features that this cell should have is the ability to divide, and to divide the membrane have to deform and split into two. So again, understanding membrane mechanics is very important. Also to deal with disease, for example, for drug delivery vehicle, I think everybody have had experience with the fiber, for example, vaccine, that they were saying it should be kept in the low temperature because these structures they are made of membranes and they are not stable in a higher temperature. So actually to design better vaccine also is good to understand biological membrane. Also, here is the cancer cells that they are they make a big hole using a laser beam in them. And when you make such a big hole, that's a micron-sized hole, in typical model membranes, model membrane, I mean by membranes that are only contain lipids and they are artificially made in the lab. In the model membrane, this will destroy the membrane. But here in the real cell, they quickly reseal, and then the cell again functions. So that's actually a problem for treating the cancer cell using, or to destroy this cancer cell, because they are very good at repairing themselves. And last but not least, also, is membrane gives you a very good material to investigate physical principles, like they are two-dimensional fluid, and you can study the formation of different shapes. You can study hydrodynamic in them in a two-dimensional systems. As a physicist, there are two areas that is very interesting to investigate. There are more, but my favorite part are these two. One of them is lateral organization. This membrane exhibit different phases. For example, in a very low temperature, they go to gel phase. In a high temperature, they go to liquid phase, liquid disorder phase. And more interestingly is that if you have a three component, that one of them is cholesterol, then you have two phases that coexist and both of them are liquid, which is called liquid order, liquid disorder phase coexistence. And these are the examples that are obtained on model membrane. So you see each color shows a different composition of the lipids in the vesicles. And membrane shape is very interesting. For example, they are affected by osmotic pressure change. They are affected by protein composition, and also the relationship between the composition and shape exists. And investigating this also is interesting. And these two, although they are done on in a model membranes, but they are also relevant for plasma membrane or biological membrane in general, because, for example, it is known that plasma membrane is heterogeneous and we do not understand the physical principle behind it. There are many different models, but none of them are strongly supported by experiment yet. Okay, the way I investigate membrane-related processes is using computational methods. So what I do usually, I use all atom molecular dynamics simulation, which is atomistic. Every atom is represented by a single particle, and then investigate one protein bound to a membrane and see the mechanic and the geometry of the structure. From this, then in the intermediate scale, I use coarse grain simulation, which I typically use Martini model to investigate how multiple of these proteins interact with one another. And then I load this data obtained, or the collective data obtained from these two simulations, and load it into a dynamically triangulated surfaces simulation, which is a mesoscale simulation, to investigate how the collective effect of this protein or any external forces on these membranes. And recently we developed a backmapping scheme, which eventually you go to from the mesoscale and go back to molecular scale. Then you can have a representation of all molecules and how they are organized in the system. So such a configuration of different multiple simulation techniques, we usually refer to them as a computational microscope, and because it has a lot of similarity to a real microscope. This is one of the recent work that we have done, was that this is a dynamically triangulated surfaces simulation representing a membrane tubular invagination, and people are not happy about it because our community is not only physicists but also biologists, that they wanted to see the real thing. So we developed this scheme to go back and see the organization of molecule exactly there. And also there could be in our modeling, we may have done a not mistake, but maybe our model are not perfect. So when we go back, we find all the problem and then we can return and then fix them. So this backmapping is not really a magic. What we do is eventually when you have this triangulated surface, increase the number of the point on this triangulated surface in a way that still the geometrical property of the original surface and the new one matches. For example, we have to keep the curvature at each point again the same thing. Otherwise, because these shapes are obtained from the Halfrich Hamiltonian that are based on the curvature, and the new surface should not have a different curvature from the original one. And from this scheme, we could do it, and then we could add protein and everything and go backward. Okay, this is one of the examples that I use this multi-scale simulation to investigate. Is how Shiga toxin induces tubular membrane invagination. This is a very important process. First of all, to understand disease, one of them is this protein is a bacterial toxin. It causes diarrhea. And the way it enters the cell is very interesting. They actually exploit the cellular molecules on the surface of the plasma membrane, which is a GB3, as a receptor. And they bind to it, to 50 of them. They induce a curvature, and then they cluster, and then they induce tubular membrane invagination. But it's not clear how all this single process happens. And also to add to this, only the A subunit is toxic. The B subunit actually is like a car. It's delivered the A subunit into the cell, and it can be used for drug delivery vehicle, because if you install anything on it, it still enters the cell. So you could install drug and then go to a targeted place. So our investigation is only in the B subunit, and this process doesn't really change if you have the whole complex or only the B subunit. And yeah, so we run multi-scale simulation using all atom molecular dynamic. We find curvature, for example, and the mechanical changes in the membrane. Then we run, here is DPD, is not Martini model, but we run DPD simulation to investigate how these two protein cluster. And it's very interesting. This is our conclusion based on experiment and simulation was that this is as a thermal Casimir-like force. Is not it doesn't arise from the quantum fluctuation, but it arises from the fluctuation of the membrane shape. And then when we load these two informations into these dynamically triangulated surfaces, you see that this tubular invagination forms, which is very close to what you see in experimental setting. And is more interestingly, if you don't have this information about, for example, mesoscale, the protein failed to actually induce the tubular invagination. So you somehow need all the scale to capture the realistic process. Here we also, based on this, we hypothesized that this is the thermal Casimir-like force. So what we said that if this protein can somehow remove their ability to suppress membrane fluctuation, what happened? They should not cluster anymore. And we add this kind of linker to stop them from suppressing membrane fluctuation. And what we see is that as the linker size increases, they separate. And then this allowed the experimentalists also synthesize this lipid. This is a GB3 with the additional linker in between. And they see actually both clustering and the tube formation is affected as the linker size increases. Okay, and then another one was that, yeah, let's add more experimental data into computer simulation, especially molecular based computer simulations. And what we did, this is the realistic mitochondrial structure obtained from cryo-EM electron tomography. So we could triangulate this density map that is obtained from experiment, and then using this software, this scheme that I just talked about, you can increase the number of the point, and then you can add molecule to it and actually run a realistic size. Only the size is realistic, a simulation of entire mitochondrial system. Yeah, and then another one was to simulate COVID-19 envelope. So this time I used a top-down approach. From experimental data, I set up this mesoscale simulation of the protein on a membrane, and this is the line representing M dimer, and then the blue, red dots representing the spike proteins, and then you can run it and you see the organization of the this protein on the membrane. Then we back mapped it into near atomistic resolution simulation, which is very complex system because and also solvent are present here. Just I remove them for clarity. And you can see the internal part is highly covered by protein, and it's very numerically also is very difficult to build such a structure because they are often unstable. And we built this and we ran molecular dynamic simulation so we could get a realistic organization of the envelope of COVID-19 virus. Yeah. And so it's not only about producing biological systems, this multi-scale simulation or this method. Sometimes you can also investigate physics, actually, or certain phenomena that you obtain from theory, and then you wanted to check it in more complicated situation. For example, here is a very nice example. I think it's already 3 years old, so that's why I didn't want to talk more about it. But what happened is, imagine you have a bilayer. This is in a periodic box, so just a flat surface, and you have protein that induces membrane curvature. Theoretically, you can predict that based on stability analysis, only in a linear regime, that the effect of this protein is only to reduce the effective bending rigidity. So it scales like this. And then there will be a point that eventually bending rigidity will become 0, and then it will form a branch polymer. But we did these simulations on different concentration for different membrane curvature. What we see, it's actually true that it becomes softer and softer as the curvature increases. So do the also the coverage. But what happens is for high curvature, there is a certain concentration that suddenly it goes to 0. Suddenly the bending rigidity go to 0, not to 0 because it become undefinable. It's not reasonable to define it anymore. And the reason is when the membrane becomes so soft, the fluctuation of the shape dominates, and then suddenly you see a phase segregation of these proteins. Because of this high fluctuation, you have a very highly curved shapes, and then suddenly protein that induces curvature goes to this highly curved part, and you have a domain with highly covered and highly rich in the proteins, and a domain with lower concentration of the protein. So you see some sort of phase coexistence that is shape and fluctuation driven. By the way, you want, if you increase the curvature or the concentration farther, especially the concentration farther, this effect actually reverse. It's clear from this equation if theta is the if theta is the concentration, and we see this again. So this is not mechanical property, but as thermodynamic, both fluctuation and the mechanic both are involved. Okay, so just to remind you, what do I mean by TS2CGs was this scheme to backmap, and now we are working on it to make it better and to be able to include more experimental data, like here from electron microscopy. We wanted to create exactly the membrane mimicking this shape from the figures and building such a structure, so eventually you just observe something in experiment, you use simulation from this structure and create a dynamical view at the pico-Newton time scale and nanometer length scales. Here is also another example, highly curved and complex shapes that can be obtained from this scheme. And now we are actually going toward simulating a minimal cell for now. A minimal cell, which is this minimal cell exists, so and already has been a simulation of a kinetic model-based simulation of this, which they see the division using this kinetic model. But now we want to have a simulation at the molecular scale. So yeah, the TS2CG plus the polyply developed by one of my colleagues allow us to both put the protein and the ribosome and the DNA into the structure, and also create membrane with any composition and shape into it. So eventually we aim at simulating the entire mitochondria. This time we want to include the protein and make a realistic simulation, and then also simulating the entire minimal cell. Also, we can create membrane shape based on equation. So you just give an equation and then gives you a shape, and this allows you to run small system simulation to see. There are more lipids here. I just showed one type of it that you can see there is a phase segregation of this lipid induced by the membrane shape. So you can study this and make such a data, which eventually tells you which lipid like which type of curvature. And then from this, we can eventually use a machine learning algorithm or different type of algorithm to set up a proper structure very close to equilibrium, and then run simulation to have a look at the dynamic of view. Or we can also run free energy calculation and obtain the relation between the lipid configuration and membrane shape. Okay, so this will be the last part, and I want to talk about this synthetic membrane shape that is in collaboration with Delft University in the Netherlands, and so this is the main people involved in this project, but there are more, so you can look at the archive version of the paper for other people. And this is a part of a consortium in the Netherlands to build a synthetic cell from bottom-up approach. So eventually just take all the building blocks, put them together, and see if you can create a cell. And one of the properties this cell should have, this is the definition of for the success of the project, is that this cell needs to be able to divide. So therefore, we want to try to make a division machinery for this, how to control the membrane shape and then split it. This work is only how to control the membrane shape. So they designed this DNA-based structure, which is like a nanostar, and they are connected to a cholesterol molecule through a single-stranded DNA. And then what they see is that this structure can create these stomatocyte structures and also this dumbbell-shaped structure. Depending on the concentration and osmotic pressure, you could obtain this kind of. So you, with using this structure, you can control the membrane shape as you want, or at least the possible shapes. And this one is very important because then it allows you to design a division machinery. You can, and then experimental observation was when they, okay, just to make this clear here. So this structure happens when you have the, this nanostar outside. No, sorry. When you have this star structure inside, and here you have it when you have it outside. So one side only has the nanostar in it, and then what they see is they could also, because nanostar is only in one side, so other protein can bind from the other side. And when they have this, the nanostar are inside, and then dynamin is outside. So the dynamin is a protein that can cut the membrane neck. So this necks, here is this tiny necks here, and then this dynamin is capable of cutting these necks if they are activated. Here they are not activated. But what is interesting, when you have dynamin outside, they go to the neck. And also when you have a dumbbell shape as well, and the dynamin is inside, you still see that dynamin goes to the neck. These are dynamin. FTZ is also known to be contributing to bacterial division machinery. So for dynamin, for FTZ also, it worked in the same way, both concentrate in the neck, but apparently FTZ also is capable of extending these necks more. And from this observation, we wanted to actually develop a model for it, and also to understand how this nanostar induces the membrane curvature. So what we did was, okay, before going any further, let's now come back to the detail of the method that I was doing. So the method for this is dynamically triangulated surfaces simulation, which is mesoscale simulation. And in this model, each surface is represented by a triangulated mesh. Each membrane is a triangulated mesh, and each vertex in the model represent a patch of membrane, a patch that contains hundreds of lipids. So it's quite coarse grain. Using a certain discretized algorithm, we can obtain geometrical properties of this surface, like mean curvature, Gaussian curvature, and an area associated with. And we have this shape operator based approach. Also, you can obtain the directional, also principal directions associated with the maximum curvature and minimum curvature, and also the normal vector. So you can obtain a local coordinate at each vertex. So when we have this property, you can use a discretized version of half-free Hamiltonian, which eventually is just a harmonic potential of mean and Gaussian curvature. Now, of the principal curvatures, not the Gaussian. Gaussian curvature is already second-order term. Then, based on this model, a membrane is represented by three properties. One of them is bending rigidity, Gaussian rigidity, and a C-node, which is a spontaneous curvature, meaning that if the surface has the tendency, due to its material property, to bend toward a certain direction. And this Gaussian curvature term is usually constant unless the topology of the system changes, which is not happening in most of the cellular processes, so we really can ignore this term. And the way system is updated is through Monte Carlo simulation. Each vertex is moved around, and then you accept or reject the move based on metropolism algorithm. And to give a dynamic to the system, also a link should flip. So vertex move and a link flip gives a fluid character to this surface. So it's not a static surface. It's not like a paper. It's a fluid surface. Okay, so based on this model, and then in connection with the experimental setting, we need a term to also control the volume of the system. So because of the osmotic pressure, meaning that inside and outside have a different concentration of the ions or certain other solvent molecules, and a pressure will be induced that want to equalize somehow the concentration in both sides. So this the osmotic pressure to a good model for it could be just linearly dependent on the concentration of the solute. And so therefore the osmotic pressure difference will be one of them from outside, a concentration from outside, and a concentration inside, and this one will be volume dependent. As the volume gets smaller, the concentration increases because the number of the particles inside the vesicle are constant. So from this we can obtain equilibrium volume that osmotic pressure difference is zero, let's say. So this will be, and then integrating this equation to obtain the energy associated with the volume change, we get to this equation, and then we can get to something like this, which eventually, depending on this parameter, this term, we can say which term is important, and this term is very big, is for the current system that we are studying, because the concentration is in a millimolar and the volume is in hundreds of micrometer. So this energy will be very larger than this number, very larger than hundreds of kBT, which is the energy associated with membrane bending. And therefore we can ignore this term and say, okay, membrane go to a fixed volume and then only fluctuate around this volume. The volume doesn't change. So we just couple our simulation to such a potential, and we know is the volume of a complete sphere with the fixed surface area. And then there is small v is somehow defined, tells you how much volume is reduced. So only input parameter will be this null, this v is small, and then the kappa, the rigidity or the coupling parameter, which we usually use a big number because in this experimental setting, this is the big number. So based on this, we can see the transition. These are known results. I mean, this is not new. So based on this, we have transition from a discoid to stomatocyte structure that was already also predicted by theory, and then you can see other type of structure that can be obtained based on if you have spontaneous curvature and negative or positive, and depending on the volume or reduced volume of the system. And they are quite well mimicking what we see in the experiment. This tells us that, yeah, because we need, first of all, just makes one more thing clear, is that without spontaneous curvature, you always see stomatocyte structure. You never see a dumbbell shape. So therefore, somehow tells us this nanostar should actually induce curvature from this result, because based on these two parameters, we could get exactly what we get in the experimental setting. And then we turn to molecular dynamic simulation to see how this actually works, if it induces curvature or not. So we just model the cholesterol covalently bound to the single-stranded DNA, not the whole nanostar, just to see what is happening. What we see immediately is that the area per lipid of this system compared to cholesterol increases significantly by 10%, meaning that the area here has increased compared to here, somehow induce a wedging effect, creating a curvature, membrane curvature. And this also was clear from the radial distribution function, as we have the nano. This is a system containing cholesterol oligonucleotide, which means the single-stranded DNA part. And this one is just cholesterol, and you can see the cholesterol has the peak of cholesterol, the first peak of radial distribution function for cholesterol is very high compared to cholesterol oligonucleotide, and this is the radial distribution function of cholesterol with respect to POPC lipids in the system. So with respect to other molecules, meaning that cholesterol oligo distance with the other molecules is larger and making it some inducing and wedging effect for the curvature. We also define this positive curvature index, which eventually tells you whenever your molecules are in a positive curvature region, is 1, and if it's not, is 0. And then what we see is that it's actually larger than 1 when you average over the simulation time, while the same is not true for the cholesterol. Is actually lower than 1, which means it induces negative curvature, or it prefers negative curvature. So we have a good understanding actually how this system induces curvature, and it's through this small part, this single-stranded DNA part that get embedded into the head group region of the membrane. The rest is not really that important or has a minor effect. So, okay, and then let's go just to show you next. Next is to have a look and see how this protein actually localizes in the membrane neck, at the membrane neck. And for this, we have to make a protein model for this dynamically triangulated surfaces simulation. And a protein model, a protein is modeled as an inclusion or a vector on a vertex that can jump from one vertex to another. And then it has also orientation, in plane orientation. So in the plane of the vertex, it can rotate. For elongated protein that induces elongated curvature profile. This is a curvature profile. We model it as we add this term of the energy to every single vertex energy, and it has 4 model parameter, K1 and K2. K1 somehow tells you how much energy, if the curvature preferred with this protein is this C0 number, if the membrane deviates from that curvature, how much energy penalty exists for it. And this one is parallel to that one. This is for parallel to that orientation, and this is for the perpendicular to that orientation. This one, this C parallel and perpendicular are protein membrane curvature in the direction of the protein. So somehow we have to use Euler formula to obtain curvature at the direction in the direction of the protein, because we only have principal curvatures. And if your protein inducing isotropic pressure curvature, the more general form of this up to the second order, the more general form of protein model is like this, which has only 3 model parameters. One of them is induced curvature, an increase in the bending rigidity, and also an increase in the Gaussian rigidity. And then we have to also include protein-protein interaction. And these protein-protein interaction are a bit tricky, because on a curved system, we have to translate the orientation of these vectors to the in the direction of geodesic distance between the vertices that protein are on, between the 2 vertices. So when the 2 vertices are non-neighbor, we just use a 0 potential. So it's a very short range potential. When they are in each other neighbor, we use a potential like this that depends on the orientation of each protein in their plane, but after the parallel transport. So the angle between the 2 proteins will be obtained after the parallel transport. And what we see is that if we use the first model for protein with elongated protein, we get 4 situations depending on if the protein are from inside inducing positive curvature or negative curvature. We get this 4 profile, which is actually mimicking the experimental situation quite well. But just to clarify, we have to have such a aggregation potential to get this, otherwise they will not just go to the next. So it's a collective effect. Single molecule really, single protein really doesn't do much. They have to collectively collaborate with each other and go to membrane neck and then actually change the structure of the neck. If we use the other model, the symmetric model of the protein, we don't really get proper result. First of all, the protein do not go to the neck, or sometimes even they induce their own invagination and they cluster into the invagination stomach, not the neck actually, or even in the case of dumbbell shape, they still don't do that. So we just said possibly these proteins are not symmetric. So they will inducing, or at least maybe they inducing filaments or anything, but they can't be properly modeled as a symmetric inclusions. And if we compare this to the experimental setting, we see that, yes, here is the next getting extended and the protein are aggregated around it, which fits very well to the FTSZ case. This means that FTSZ induces negative curvature. Wait a minute. Yes, and then this is for and they are consistent. So protein positive curvature binding from outside will be like this, which somehow shows that case for the dynamine. And then dynamine also induces again positive curvature. Even if they are binding from outside, they still do not extend the membrane neck, while the FTSZ case, they still they extend the membrane neck. In all 4 cases, they go to the membrane neck, but depending on positive curvature or negative curvature, they either extend the neck or no, they just stay there. Okay, thank you very much for your attention. And here are the people that I have been working with for these results. And yeah, that's it.
Thank you, وریا, for this interesting talk.